Alzheimer's and the amyloid hypothesis. Alzheimer's disease is a progressive disease in which dementia symptoms get worse and worse over time. Some of the symptoms are memory loss and confusion. Alzheimer's disease is named after Alois Alzheimer. He gave a lecture in 1906 on one of his patients, Auguste D., who was a 51-year-old woman, and she displayed symptoms of dementia and cognitive impairment. In his notes, he wrote, "When objects are shown to her, she does not remember after a short time which objects have been shown. Asked to write Auguste D., she tries to write misses and forgets the rest. It is necessary to repeat every word." So basically, she was showing symptoms of memory impairment, which are usually the first and most pronounced symptoms of Alzheimer's. After she passed away, an autopsy was performed, and plaques and neurofibrillary tangles were found in her brain. This, in essence, gave birth to the amyloid hypothesis. It's interesting to note here that Auguste D's age, 51 years old, because of that, it was thought that the disease Alzheimer's was pre-senile, or in other words, is disease of the young. This might explain why there's a gap in the research on Alzheimer's, and most of the work has been done in the past few decades. Alzheimer's disease is characterized by the accumulation of beta amyloid peptide and tau protein. Today, we won't be taking a look at tau protein, and we'll be focused just on the beta amyloid peptide. In the picture below, you can see the neurofibrillary tangles that form within the cell body, and these are caused by tau protein. Amyloid plaques are on the side, they're outside of the cell body and are formed through the accumulation and aggregation of beta amyloid peptide. The amyloid hypothesis revolves around the idea that Alzheimer's is caused by the accumulation of beta amyloid peptide. In the picture on the right, we can see another amyloid plaque, and this was, I believe, done through immunohistochemical staining, which is a good and easy way to look at amyloid plaques while doing research. The beta amyloid peptide is formed from the sequential proteolysis of the amyloid precursor protein, APP, and we'll be taking a look at how that happens exactly in the next slide. It's interesting to note that we don't know currently what amyloid precursor protein does in the brain. In studies with mice over expression of APP, positive effects were shown on health and the growth of cells. In this slide, we'll be taking a look at how amyloid precursor protein gets turned into beta amyloid peptide in the normal brain. In the normal brain, alpha secretase cleaves amyloid precursor protein into secreted amyloid precursor protein, SAPP alpha, and the C-terminal fragment, which is membrane-bound and 83 amino acids long, CTF83. Then gamma secretase comes along and cleaves CTF83, producing APP intracellular domain. AICD makes its way to the cell nucleus where it functions as a transcriptional factor. In an Alzheimer's brain, amyloid precursor protein is cleaved by beta secretase, generating secreted amyloid precursor protein beta, SAPP beta, and a C-terminal fragment, also membrane-bound, but this time with 99 amino acids. CTF99 is also cleaved by gamma secretase. This also produces APP intracellular domain. However, it also forms beta amyloid 4042 peptide. Beta amyloid 4042 peptide then interacts with the polyprotein E, which allows for the aggregation of beta amyloid oligomers, which leads to amyloid plaques. Plaques are insoluble and dense and are found outside of the neurons as shown before. So how exactly does the aggregation occur? The beta amyloid monomers themselves are harmless, but at higher concentrations, they undergo a conformational change, which results in a tertiary structure rich in beta sheets. So what I'm drawing here are oligomers, which have been formed by monomers, which were partially unfolded, misfolded, or structurally disordered, and began to interact with each other. These oligomers then begin to interact with each other, and they begin to form a nucleus. This step is called nucleation, and it is the rate-determining step in the ultimate formation of fibrils. This phase, however, is highly energetically unfavorable and allows for the further addition of monomers at a greater speed. This is called elongation. Lastly, we have the formation of the amyloid fibrils. which are rich in beta sheets, and this is a highly favorable position in comparison to nucleation and elongation. These fibrils are then deposited as plaques, which prevent things such as neurotransmitters from being released, blocked ion channels, and dysregulation of glucose and metabolism. The chart on the right also shows this process. You begin with a monomer, which then begins to interact with other monomers to form oligomers, which then interact with each other to form a nucleus, which is the nucleation step, which is the rate determining step. And after this, the addition of monomers becomes easier and easier. So this is the elongation phase, which then leads to fibril formation. To summarize the overall process of Alzheimer's, we begin with the cause. This could be both aging or genetic. This leads to beta amyloid peptide accumulation, which we saw the many procedures involved. This includes the formation from amyloid precursor protein to the procedure of its aggregation. This is followed by neuronal injury, where the neurons are no longer able to function correctly. This leads to altered tau metabolism, which was not covered, but we saw in the pictures in the earlier slides how they create those neurofibrillary tangles within the neuron. This then leads to further neuronal injury. Lastly, you have neuronal dysfunction and cell death, which can contribute to the overall symptoms of Alzheimer's, which include dementia, confusion, memory loss. Amyloid aggregation features beta sheets with varying amino acids, which makes them difficult to study. In 2012, researchers designed beta sheet macrocycles which allow for the display of sequences of the amino acid and various amyloid proteins. These macrocycles are referred to as amyloid beta sheet mimics, or ABSMs. The researchers specifically generated ABSMs with inhibitory properties. They function through inhibiting the aggregation of amyloid proteins, which prevents the formation of amyloid fibrils. ABSMs function by binding beta amyloid oligomers and preventing beta amyloid nucleation. As a result, the pathway is changed and fibrils are not formed. It was found that ABSMs had a greater affinity for the early beta-structured oligomers than unstructured monomers. I understood this as ABSMs were competing with the monomers for a spot on the oligomer. The binding of ABSMs to beta amyloid oligomers creates a beta amyloid oligomer ABSM complex. This complex is stable, which results in a higher energy barrier for nucleation to occur. This barrier can be seen in the reaction coordinate diagram below. As you can see, the dotted line represents the ABSM oligomer complex, and there's a higher energy barrier needed to get to the nucleation stage. To understand how the ABSM may complex with a beta amyloid oligomer, it is important to look at the structure of the ABSM. ABSM is composed of an upper layer called the recognition strand and a lower layer called the blocking strand. Together, they form the monomer ABSM. The upper layer is known as the recognition strand because it contains the peptide found in beta amyloid. It is composed of 7 amino acids, which are all hydrophobic. It's called the recognition strand also because it is able to bind the beta amyloid oligomer. The lower strand is known as the blocking strand because it functionally prevents beta amyloid from aggregating. It does this through the use of this HAL unit, which is presented right in the middle. It's 3 peptides long and functionally prevents hydrogen bonding. Basically, it lowers the amount of exposed hydrogens, which removes hydrogen bonding capability. While the upper strand is bound to the beta amyloid oligomer, this allows the lower strand to prevent beta sheet stacking and aggregation, which in turn inhibits fibril formation. It's important to note that the ends of the monomers here are linked together by ornithine, which is a non-proteogenic amino acid. It is used here because it allows for stabilized turns. Studies have actually found that ornithine allows for more stable turns than asparagine and glycine. This beta sheet is anti-parallel because the amino acids in the upper and lower strands run in different directions. The upper and lower strands interact with each other through hydrogen bonding. The hydrogen bonding between the upper and lower strands occur between the carbonyl oxygens and the amine hydrogens, as shown here. In the next slide, we'll take a look at which amino acids compose the upper and lower strands. All of the amino acids in the upper strand, or the recognition strand, are hydrophobic. The first amino acid in the upper strand is alanine. The second two are isoleucine. Then there's glycine, leucine, methionine, and valine. The sides, as I mentioned before, are ornithine. These 7 amino acids are essential in being able to interact with the beta amyloid oligomer. In the lower strand, there's only 4 amino acids, which excludes the Howe unit right in the middle. The first amino acid from left to right is lysine. The second one is phenylalanine, and then this repeats with phenylalanine being the third and lysine being the fourth. Together with the Howe unit right in the middle, the lower strand prevents aggregation of the beta amyloid oligomers and thus prevents fibril formation. This slide shows the ABSM structure in 3D. I've labeled all of the amino acids right here for the upper and the lower strands. As shown before, this is ornithine right here, and this is the Howe unit right in the middle. And I'll play this video clip of the ABSM structure, which will spin around so you guys can see what it looks like. This slide shows ABSM in crystal dimerizing with itself. This was done to show how ABSM might function when it encounters a beta amyloid oligomer. When ABSM complexes with beta amyloid oligomers, it is thought that edge-to-edge hydrogen bonding interactions between the ABSM and the exposed hydrogens of the beta amyloid oligomers occur. We can see that there are edge-to-edge hydrogen bonding interactions between the 2 ABSM units. Additionally, the upper strands interact with each other through hydrophobic interactions, and while the upper layers interact with each other, the bottom layers do not, which prevents aggregation. This then prevents the beta amyloid oligomer from reaching nucleation, which thus prevents fibril formation. It's possible that in the future, these ABSMs may be used to treat people with Alzheimer's.